I'll let you do it. And blame me for that one.
Okay, can everybody hear me? So I'd like to thank the organisers for inviting me. I'm going to try to pack two stories into my talk today. They're both related. One of them is in the abstract and the other one is completely unpublished. So I want to talk about a different approach to structural variation, which you can get from low coverage sequence of population data. So most of my career in genetics has been based around what we might call multi-parental populations. So you start off with a bunch of inbred lines, and you cross them according to some scheme, and you may end up with a bunch of new inbred lines whose genomes are mosaics of these founders, or maybe you'll do some kind of outcrossing scheme where you end up with heterozygous mosaics. But nonetheless, the assumption is that everything is just a mosaic of the founder haplotypes. And if you know the mosaics, you know everything. Okay? And to some extent, that's implicit even in natural populations. You know, the whole HapMap project, the human genome is based upon the assumption there is a limited space of haplotypes. Everyone is a mosaic of those haplotypes plus a sprinkling of rare, possibly private mutations. So I've decided to call this a central dogma just to be irritating. The question is, how true is this statement? I mean, it's obviously true to a large extent, but are we missing variation which is not captured by this sort of mosaic view of the world? So, first part of the talk is going to be on the Arabidopsis, which is not fruit fly. So it's a highly variable relative of the cabbage. These are just different accessions grown under different environments. You can see great differences in size. And it's a naturally selfing species, so it makes inbred lines very easily. So the population I work on is called a MAGIC, which stands for Multi-Parental Advanced Generation Intercross or something. I can never remember what it does stand for. So we have 19 founders for this population, and this is sort of, the breeding scheme is shown here. So on this side we have a diallele cross of all 19, and the idea is you then pick a pair of F1s to make an F2, and then you'd pick a pair of F2s to make an F3 until you get down to an F4, which should contain something like, you know, well, it could contain 16 parents. It usually contains about 12. And then you self it for 6 generations, which is enough to make it essentially inbred. So you end up with genomes looking like this. So this is the output from a hidden Markov model run on SNP genotypes. This is one individual, magic line 100, and we have the 19 founder genomes here, and what we're plotting is the posterior probability that this individual is from this particular founder for that particular bit of the genome. And you can see over most of the genome, you get very, very clear identity. Okay, so the haplotype mosaic for chromosome 1 is pretty well defined. Over here, it's a mess. This is a centromere of chromosome 5. And the question is this a mess because you have 2 segments which are identical by state, or is it a mess because there's something more interesting going on? So we kind of revisited this population and sequenced a large part of it at low coverage. This was done some years ago. It took a long time to get published. So eventually the paper came out in 2017. Martha Impriou did nearly all of this work as her thesis. So the approach we took was we wanted to look for things other than SNPs, so that primarily means some kind of structural variation. If you have short-read sequencing data. It comes in read pairs, and there's a large cottage industry of software developers writing programs to try and call structural variants based on these short-read data. So you're looking for essentially read mapping anomalies. If the read pair maps to the genome properly, you'll have two reads pointing together at a given distance apart, give or take. But if you have them both pointing in the same direction, for example, that indicates there may be an inversion. If they're further apart than they should be, that indicates it's probably a deletion. If they're on different chromosomes, possibly some kind of rearrangement. However, that data is incredibly dirty and messy, and it's generally accepted to be extremely difficult to actually call structural variants accurately from low coverage sequence data, unless you have a lot of data, which we didn't. So we took a different approach, a sort of more statistical approach. We divide the genome up into bins. Each bin is 10 kilobases long, so that's only 12,000 bins in the case of Arabidopsis, or indeed Drosophila. I could give this talk on Drosophila. I've got confused. And then what you do in each individual is you count how many bad reads there are mapping to that particular bin in a particular individual. Okay, so you're interested in the bad stuff, yeah? And that will vary from individual to individual. Okay, so you know, in some individuals, their haplotype at that particular locus will be the same as the reference, so everything should line up nicely. Other individuals may contain a structural variant there, so you would expect to see an excess of anomalously mapping reads. And in fact, there are many different measures you can use. We ended up using about 6 different measures. It doesn't really matter what they are, particularly. What we have down here is chromosome 1, and we're plotting the variance of the numbers of badly mapping reads in each bin across our population. And you'll see that, generally speaking, it's at a constant level. That's partly because we quite heavily normalized the data to start with, so there aren't any artifacts as far as we can tell. But you get some bins where you suddenly get a big spike of variation. Okay, so there's a lot more variation at some bins than in others. And the other thing you notice is that these bins tend to be fairly isolated. Okay, the spikes are isolated. In the magic population, there's a lot of linkage to equilibrium. You know, it extends over about 200 base pairs. So the patterns we're seeing here are quite distinct from those that we would see if we were looking at SNPs. This is the centromere, which obviously has a lot more structural variation because it's full of repeats. So the idea is that where you have a bin with a lot of variation, that indicates what we call a structural variant trait. And you can then map that genetically. And the logic goes like this. Supposing back in time, there's an individual, and this segment in this individual moves to a different chromosome. Okay? So we now have a breakpoint on the original chromosome and an insertion over here. So it's going to land in a particular haplotype context. Now let's think about what we might see in the present day population, which is descended from individuals, including this one. And for simplicity, let's suppose that the original state here magically becomes the reference genome, okay, for just for simplicity. Then because recombination and random assortment of chromosomes will occur, we'll see essentially all combinations of these these 4 states. Okay? We will see, then we sequence our population, and we try and map our genes, our reads to the reference genome. Okay? And then we will see bad stuff mapping to this locus if it's carrying the recipient haplotype over here. Okay? So, in other words, you'll get a correlation between a haplotype state at this locus and the amount of bad reads mapping here. So we can use this as a way to map structural variant traits as if they were gene expression traits, for example. And in particular, we can then distinguish between essentially local events where maybe a segment's been inverted, so we'd call that a cis SVQTL, and transpositions where the segment's moved somewhere else, or maybe it's even been duplicated. I mean, it can easily be duplicated just because of the way assortment of chromosomes works. You know, it may have been moved in the original mutation event, but then you've crossed back in the original state, so you end up with two copies, for example. Okay, so if you do this in that population of Arabidopsis, you get about 6,500 distinct SVQTLs, and about a quarter of those are transpositions. Okay, so it's quite a high fraction. This is just a subset of them. So what I've done here is taken one of the 19 founders of this population, which is L0, Landsberg, and I've just drawn those structural variants which are which relates this particular founder. So you can see the sort of patterns of movements between chromosomes, and you can see how the centromeres, which are these things here, tend to dominate. Okay. You can use this in two ways to look for association with phenotypes. So I'm going to give you a nice story first, which is very easy to interpret. So here we're looking at germination time in these Arabidopsis lines. The orange line here is the GWAS you'd get for germination time, and it has a QTL over here, which is genome-wide significant. You can also forget all about SNPs, and you can just say which of my structural variant traits are most highly correlated with variation in germination time. Okay? So in other words, you're treating the structural variant trait as like a genotype. If you do that, you get this figure here. Each spike is one bin, and those bins, which the height of the bin is the log p of the association between variation in this bin and germination time. And you can see there's a bin here, which happens to be very close to the peak here. If we plot that, if we then do a GWAS for this structural variant trait, we see this green curve here, which is essentially the inflated version of the orange curve. And if you regress the structural variant trait out of the germination time, you essentially ablate the QTL. Because this population is descended from a known set of founders which have been sequenced at fairly high coverage, we can then look at the read coverage in the 19 founders, and this region here corresponds to this bin. Okay, these are the 19 founders. And you can see, in this case, is a rather well-defined deletion of 3 genes, okay, one of which is a fairly strong candidate. So this looks like a very nice example. And this is structural variation which was clearly segregating in the founders, okay? So I think this is fairly standard story. If we now look at another quantitative trait, this is for resistance to fungal infection. I forget which fungus it is. You get essentially the same story, except it's a lot more messy. You in this case, you can reduce but not completely ablate the QTL by regressing out the top structural variant trait. And if we look at the variation in the founders, we can see that although there is some evidence of structural variation, it's extremely messy. We can also look at things like gene expression, and for a subset of these transpositions, we can actually map the breakpoints. We can't do it in most cases because, well, essentially these things are probably driven by mobile elements, and it's extremely hard to design PCR primers across the breakpoints. But where we can, we then know exactly which genes are being moved around the genome, and we have some gene expression data on a subset of these lines, and we can ask what happens to expression in lines where segments being moved around. So, gene density in Arabidopsis is quite high. It's about 1 gene every 10 kb. So you can easily have, if you've got like a 50 kb piece being moved around, you can easily have, 5 genes or something. And the sequence context of those genes appears to be intact, right? It's just that they're now in a different place in the genome. So the graph to look at is this one here. And what we're plotting is the fraction of transcripts with no expression. So outside of our structural variants, the fraction is very low. If you're entirely within, it's about sort of nearly 30%. If you're actually on the breakpoint, it's very high, but then I don't think that's a very surprising result. I mean, if you break a gene in half, it's probably not going to do that well. But this does raise the interesting question of allelism. I mean, if you've moved an entire segment around, but you've kept it intact, but nonetheless the expression is completely changed, then you need to be quite careful. If you don't know that's happened, right, you're going to treat these two things as being allelic, as being the same thing, but they're not. At least I don't think they are. We also try to map, we try to compute the equivalent of heritability for these structural variant traits. So we can define a kinship matrix just in the same way that you would do from SNP dosages, except here we're working out the correlation coefficient between every pair of lines based on their structural variant traits across the genome. Okay? So we can get a kinship matrix in just the same way that we would do if we were looking at SNPs. We can estimate these variance components, and we can use maximum likelihood to find the estimates for heritability. Now, I'm plotting here the likelihood curves, rather the negative log likelihoods, for both SNP and haplotype-based kinships, and then a variety of different structural variant-based kinships. In some cases, you can get heritabilities which are quite a bit higher than the SNP or haplotype-based, which tend to give you the same answer. But notice that the height of the curve is quite different, okay? And that's related to the fact that the spread of values in the kinship matrix defined for structural variant traits is radically different from that that you get from haplotypes or SNPs. So down here, I've calculated a kinship matrix either using SNPs or using haplotypes. So here, this just means an IBD kinship matrix. And the correlation is pretty good. So each dot is one element of the kinship matrix. If I compare, say, SNP-based kinship matrix to large insert size, which is one of the anomalies, the correlation is still present. It's 0.3, but it's much, much weaker. And this means that these curves are rather different in character, and it's rather difficult, therefore, to fit a model with both types of kinship matrix in the same time. That's a technical detail. What is interesting is that if you estimate heritability for a variety of different growth traits, the SNP and the haplotype estimates are, on the whole, pretty comparable. And then here we have different estimates of heritability associated with different types of read mapping anomalies. In some cases, these can be quite a bit bigger than the SNP or haplotype-based things. So we're picking up variation, which seems to be relevant to the phenotype, which is not tagged either by haplotypes or by SNPs. And so the haplotypes are the things which tell you about the mosaic, yeah? So this is saying, even if you know the mosaic, you don't know everything. So at first I thought this had to be somatic mutation in these lines, but actually it really can't be. Firstly, we measure our phenotypes across, say, 10 individual plants and take the average. The plants that were actually sequenced were not from that same batch. They were at least two MIUCs distant. Okay, so whatever it is, it's heritable in the sense that you can see it in different individuals from the same inbred line, okay? But nonetheless, it's not well tagged by knowing the haplotype mosaic. So my hypothesis as to what's going on is that there's some kind of lineage-specific turnover of mobile elements, which then gets fixed within each line, but tends to reoccur at the same places in the genome. Okay, so that way you get a particular place in the genome with a lot of variation, but that variation is essentially unique to each line. Okay, so I think that's what's going on. But I don't know. And the way to test this, of course, would be to resequence the population with long reads, which I'm trying to get the money to do. Second example I want to talk about is depression in humans. So this is from the Converge study of major depressive disorder. So this is based on sequencing about 12,000, well, 10,000 after QC, Chinese women, half of whom had major depression and half who were controls. They were the samples were collected from hospitals across China. And the mean coverage of each individual was about 1.7x. So this is saliva, cheek swab DNA. So if you do a standard SNP-based GWAS of this data, imputing sequences, SNPs from the sequence data, you get two genome-wide significant hits, which can be replicated in a different sample. One of them is for close to a gene called SIRT1, which has some mitochondrial association. So yeah, we find signals, but we don't find very many. This is what you get if you try to associate depression with variation in these structural variant traits. You get a very large number of potential associations, okay? This is the threshold that you would normally set for genome-wide significance. Now, the question is, are these artifacts? Okay, so I'm going to try to present the evidence that maybe they're not all artifacts. First thing is, when you do a GWAS, you have to control for population structure using a kinship matrix derived from the structural variant traits themselves. Okay, if you use a SNP-based kinship matrix, the QQ plot looks like this grey line here, and is clearly highly inflated. The number of bins that you're testing here is about 300,000, I should say. That's the number of 10 kb intervals in the human genome. But the QQ plot, when you control using the correct kinship matrix, is fairly well behaved, up to about log p of 3. The kinship matrices are very different in character, just like they were in Arabidopsis. So the frequency distribution of elements in the kinship matrix, the SNP-based kinship, is very, very tightly centered on 0, as you might expect. But the anomaly-based GRMs are much more widely spread out. And there's only a weak correlation, again, between SNP-based genetic correlations and anomaly-based correlations. The correlation coefficient here is about 0.1. It's still highly significant, but it's weak. As a sort of negative control, we can try mapping a trait which we don't think should have a genetic basis, which is the number of stressful life events. The reason we measure this is because stress is a strong predictor of adult depression. I should say early life stress events. So here we don't find any associations. Okay, so I think this is a sort of negative control that it's not just due to inflation. We can estimate heritability-like measures, and for a variety of different read mapping anomalies, we can get heritabilities for depression which are rather larger than those that we see using SNPs. And again, if we look at stressful life events or a particular subset, which is childhood sexual abuse, we find that the heritabilities are low. Okay, so again, this indicates that this is not a simple artifact. Okay, we can also do some prediction. So we've done a little bit of machine learning, nothing like the extent that I think Ed has done. We've just looked at support vector machines and sort of multiple logistic models. but we can get quite accurate classification accuracy. So we can predict with 82% accuracy in our sample, okay? I should say by withdrawing 20%, training on 80%, and then predicting into the 20, if you know what I mean. Using a structural variant loci we've identified, we get about an 80% accuracy. Okay. The best we can do with SNPs is something around 60%. Okay. The most interesting thing is if we look at the genes within these 56 bins, we get a very large enrichment of genes which have a mitochondrial function. So these are nuclear genes, but they either function within the mitochondrion or they have some association with the mitochondrion. In particular, NDUF-S7 and NDUF-S8, which are part of the electron transport chain. And these are actually two of the most strongly associated signals with log Ps of about 30. Okay. Although I haven't got a plot, I looked in detail at this particular gene, and all the anomalously mapping reads for this particular locus fall within one particular exon of that gene. Okay, but interestingly, it's protective. So the more anomalously mapping reads you have near that exon, the less likely you are to have depression. And this enrichment is quite strong. It's about a 7-fold enrichment. And that's p-value of 10 to the minus 7, whatever that means. And then you can make up stories about the other genes, which I think are interesting, but when I show it to other people, they just say, yeah, well, so what? I mean, I have wasted a large part of my life, making up stories about genes. So I thought I would try to relate this back to Fisher's paper, and so I tried to read it again on a train coming up yesterday. And I think, like most people, when he got into assortative mating, I kind of gave up. But what's interesting about that paper is he didn't know about DNA. He did know about recombination. He actually mentions recombination, which he calls coupling, but he then actually dismisses it as not affecting his results at all. I don't know if that's quite true. But because he's completely unagnostic as to what is the cause of the variation, all of what I've described here kind of fits within that framework. It's a different way of measuring variation within the genome. Okay? And I think what is important is that, we've kind of got so used to thinking of, all the genetic data as being this big matrix, with noughts, ones, and twos. That we are in danger of forgetting there's a lot more variation in the genome. Some of it is, quite likely to be functional. Whether it has the same, whether it's amenable to selection in the same way, I don't know. Clearly, the data I presented here is a little bit on the flaky side. We're pushing the edge of what we can get from short-read sequencing. But I think there is a story there. I think we're missing a trick if we just think it's just SNPs and and it's just the reference. Okay. One last thing. So Sanya Fanik did all of the depression work, and Martha Imprilu did all of the Arabidopsis work. Martha's paper was published in Genetics last year after being rejected from just about everywhere else. I had possibly the nicest set of reviews and a rejection I've ever had from genome research. But at that stage, I sort of lost the will to live. I didn't fight them. But, so eventually, it came out. But, thank you.
Thank you, Richard. Do we have questions? Come on, you guys. Questions. Mike.
Forgive me if I got confused right at the beginning, but You said that you plotted where these anomalous reads were.
Yeah.
And I was expecting you to say that what you analysed was just the number of anomalous reads in a bin, but you said it was the variance. What does that mean?
Oh, sorry. Let me... Yes, sorry. Here I'm plotting the variance across the population, just to give you a picture of what's going on, but you're absolutely right. Then in each individual, I've got one number, right. It's like a gene expression value. So
the number of reads, yeah.
Yeah, well,
okay.
a normalised number of reads.
And then you treat that as a trait, and you find somewhere which predicts that trait,
yes.
A SNP or whatever.
Yeah.
And 25% of them are somewhere else, and 75% of them are where you've actually got the anomaly.
Yes, that's right.
And then, in order to look at the effect of that on other traits, what do you use? Do you actually call a structural variant
no,
genotype?
What I then do is I just do a direct association between my phenotype, say growth, germination, whatever, and variation at that structural variant trait. So I then forget all about SNPs. I'm only using the SNPs and haplotypes in order to say where these things have moved around. But say for the human example there, we didn't try to do that because mapping all structural variants in the human genome was clearly more than one person could reasonably do. So we just look for association between phenotype and SV trait.
So in a sense, it's an association between two phenotypes, isn't it?
Yes.
Yeah, okay. Thanks.
So, Richard, I found the aspect that you were getting potentially structural rearrangements at the same region over and over again. You were kind of imagining transposons doing that. Do you think I think that actually sorghum it's dwarf gene. It isn't. I think it's a slippage one that does it, unequal crossing over maybe, that recurrently creates a deletion. But do you think slippage or unequal crossing over could be doing this also, or?
I don't know. Quite likely. I have to say I'm profoundly ignorant of the mechanisms of how DNA makes mistakes, so I wouldn't want to speculate.
Okay, because when we look for retro elements jumping, they really do seem to happen a pulse.
Yeah.
But then once those things are spread around, then there's tons of opportunity for recurrent unequal crossing over events and things like that to consistently.
Yeah, I think we just have to sequence these genomes de novo with long reads and see what's happened.
I don't want to flog the Drosophila horse too vigorously here, but there's actually quite good evidence that what Ed was raising about the unequal crossing over between TEs can be detected in mapping experiments in flies. There was a paper a couple of years ago in Genetics by Scott.
Flies? I knew I should have done it in flies.
So I think it's quite a good candidate for rapid timescale changes in the structural variants. More so than I think than the actual hopping of TEs, which tends to go at a fairly low rate in most circumstances.
I just want to make a comment on this. On the other hand, I think there is a human recurrent mutation of transposable element insertion into, is it factor 9 for hemophilia?
Hemophilia?
Yeah. So people see an insertion of, I don't know where the transposon comes from, whether it's nearby or further away, but they're recurrent.
Okay. Which chromosome is that?
I think it's an X.
X.
First mapping of the human genes, hemophilia.
Hi there. Just so unpacking your last slide where you sort of have heritability question mark and thinking about other things. I mean, your structural variants are still sequence variants. Or are you talking about other epigenetic effects, or what,
No,
what
I'm
exactly
not,
are
I'm not
you...
talking about epigenetic effects. I said heritable question mark. So in the Arabidopsis case, yes, I think it is heritable because, we're measuring phenotypes in one set of individuals and we're sequencing another set. In the human case, I haven't shown any evidence that it is heritable, in the sense that if we sequence the trio, could we see that the amount of structural variation of a particular locus was following some kind of Mendelian-ish rules? That's what the question mark was meant to indicate. I wasn't claiming a role for epigenetics, but in the Arabidopsis case, if these things are driven by mobile elements, eventually these things get methylated, and so then they stop hopping around. So there is a sort of epigenetic angle to it, but I'm not making that's the main claim. Okay.
Yeah. No more questions on Aristophanes? No? Okay.
Now, there's a project.
Thank you, Richard.
I've now remembered what I'd forgotten. Is some people may want to get a certificate of attendance in order to satisfy their university's bureaucracy. We can provide those if you just email me, and I will send you one not necessarily by return of email, but fairly promptly. So if anybody needs a certificate of attendance, just email me. I won't provide them at the meeting, but I can send them out later. And I think that's it. Is Daniel Crouch here? Because we haven't got his talk on the thing. Yeah. Okay, right. Right. Well, we're a little early for the tea break, so let's hope there's some stuff downstairs. And thank all the speakers again. Well, it's a bit of a cocker. He was away yesterday. He got redirected and hit the wrong email box.
Okay. No issue.
So, a bit of fiasco. Just stick it in.
This seems to be working now. Yeah, it's not mine, yeah. How do I get to my computer? Oh, okay.